Thank you for all of the comments that we are getting here, and thank you for listening to everybody. We hope everybody is enjoying the presentations.
All right. Good afternoon, everybody. My name is Motolani Matthew. I'm a PhD candidate at the Chemistry and Biochemistry Department of the University of Arkansas, Fayetteville. I'm also a research student at the Arkansas Statewide Mass Spectrometry Facility. The title of my presentation is Folding Energy of Wild-Type Human Acidic Fibroblast Growth Factor 1, measured by protein equilibrium population snapshot, hydrogen-deuterium exchange, electrospray ionization mass spectrometry, shortened as PEPS-HDX. Now, what is this protein FGF1 and what does it do in the body? Now, FGF1 belongs to a group of proteins that are useful in the positive and negative regulation of cell proliferation, cell differentiation, cell survival, and so on. In adults, these proteins are useful for tissue repair and wound healing. Now, FGF1 belongs to the subfamily FGF1. Now, the one I'm focused on is human acidic FGF1. So the other member of the FGF1 subfamily is FGF2, which is the basic form. The two of them are prototypes, but they define their isoelectric points. So FGF1 is expressed in the retina, in the brain tissues, and in bone marrow. But FGF1 is not stable. In vivo, it requires heparin for stabilization and activation. So the first step of what happens in the cell is that heparin binds to FGF1 and activates it, and then the complex binds with FGF receptor at the extracellular end of the tyrosine kinase. And that activation or that complexation activates and facilitates the FGF1 migration through the across the membrane into the cytoplasm, where one of three pathways are activated, which could be the Ras protein pathway or the phosphoinositol 3 kinase pathway or the DAG pathway. And the result is cell morphogenesis, migration, proliferation, and so on and so forth. But the challenge is this FGF receptor is prone to mutations. And these mutations and some of the mutations in this pathway have been implicated in abnormal morphogenesis and drive or progression of several types of cancer. So the idea is to engineer FGF1 that will not need to complex with heparin. So an FGF that will bypass even the first step and can be administered directly to cells for wound healing and tissue repair, and even in the treatment of diabetes. So that is where my experiment comes in, measurement of this protein stability and the mutants that have been engineered. So the method I use is hydrogen-deuterium exchange, mass spectrometry. So hydrogen-deuterium exchange is the exchange of amide protons with deuterium. Deuterium has a mass of 2. Protons have a mass of 1. So the experiment is such that the amide protons in protein, they are the protons that are attached to the backbone of the protein, attached to nitrogen, particularly on the backbone of proteins, and I've tagged them type 2 protons. Now the type 3 protons are the protons that are attached to carbon covalently on the protein. They do not exchange. The type 1 protons are the ones that are attached to nitrogen, sulfur, or oxygen on the side chain of the amino acids that make up the protein. They exchange very fast during mass spectrometry, and they back exchange from deuterium back to hydrogen very fast also, so they do not contribute to the mass increase that we measure during mass spectrometry. So we are concerned about amide protons, those attached to nitrogen on the backbone. And these amide protons can be classified into 2 also. We have the amide protons that are found on the surface of the protein. So when we expose them to deuterium environment, they exchange very fast. But we also have those type of amide protons that are buried within the hydrophobic core of the protein. And because these amide protons, most of them are involved in secondary structure formation, they are hydrogen bonded to other parts of the protein, so they are not readily available to exchange with deuterium. So in order to make them exchange, we have to denature the protein, either chemically or thermally. So when we now increase, or when we apply the denaturants and we increase the concentration, these amide protons becomes gradually open, gradually available for deuterium exchange, and then we monitor that increase. And by so doing, we can extract the stability information of that protein. So the unfolding process follows this principle or this assumption that when a protein is fully closed, that is when it is folded, the amide protons are not available, but at the application of the denaturant, the amide protons becomes available. And so they become exchanged with deuterium, so they are labeled, and then we can measure the mass increase due to the exchange of deuterium with the amide protons. So this is the summary of the experiment. We have protein in solution. We apply the denaturant, we increase the concentration gradually, and then the protein becomes open and is labeled with deuterium. We quench the labeling step by addition of 0.2% TFA. That drops the pH to 2.7, and then we control the temperature and drop it down to 0 degrees Celsius. And that condition of pH 2.7 and 0 degrees Celsius actually limits back exchange. So the hydrogen that we have already exchanged with protons, the deuterium we already exchanged with protons are locked in the protein, so they do not back exchange until we finish the analysis. So the entire solution is injected into the liquid chromatography machine. The salting takes place, and then we'll have the mass spectrometry. So the molecular weight is displayed as mass to charge ratio in the display window of the mass spectrometer. So this is a typical spectra of FGF. I just highlighted one of the charge states. Here we have plus 17 charge states. At low guanidinium chloride concentration, which was the denaturant we used, we see only one peak in the spectra corresponding to only the closed state of the protein, because at lower guanidinium chloride concentration, the protein dwells more in the closed state than in the open state. So only a small fraction of the amide proton is exchanged with deuterium. So we see one state, one peak. But as we increase the concentration of guanidinium chloride, we can see two peaks in the spectra, one corresponding to the unfolded fraction of the protein, while another corresponds to the folded fraction of the protein. We can actually deduce the equilibrium constant value for this folding, for the protein folding, by finding the ratio of the open state to the closed state. We can also obtain the equilibrium constant by using the weighted average, which I will explain later. And then at high guanidinium chloride concentration, we only see one peak because the protein dwells more in the open states than in the closed state. So all the amide protons are exchanged, almost exchanged, with deuterium at high guanidinium chloride concentration. So this is the peak. We zoom it out to obtain the molecular weight. We use the weighted average. When we zoom it out, we have the area under each of the peak values that we have. And then we use the formula, this formula here, to calculate the molecular weight for each guanidinium chloride concentration that we have used. The data analysis is such that we obtain the observed or experimental molecular weight. These are the blue dots we have in this graph here for 3 different charge states of the same concentration and the duplicate runs that we have. And then we have the plots against the guanidinium chloride concentration. We have the increase in molecular weight gives us a sigmoid curve, but we also go ahead to obtain the calculated molecular weight, which we derived from the first principle of linear denaturation constants and the free energy change equation. So we have this calculated molecular weight equation, and then we use that for each guanidinium chloride, we use that value or the set of values to fit the calculated molecular weight to observed molecular weights using the Excel solver tool. So from there, we can extract the linear denaturation constant value. In this case, we had 5.6 plus or minus 0.1 kilocal per mole per molar. And then we had change in free energy as minus 6.4 plus or minus 0.2 kilocal per mole. Now, in order to confirm whether our PEPS value actually makes sense, we decided to count the number of amide protons in the protected regions of the protein. That is, the amide protons that are buried that will not initially exchange except the protein is denatured. So when we found when we counted from the crystal structures, we obtained 55. And then from our normally for PEPS, we use 90% deuterium environment. And then from that, we obtain 50 as our amide proton. So if we correct that, or we kind of use the correction factor back to 100% deuterium environment, that will give us 54.8 plus or minus 2.8 Dalton, which is within the range that we obtain for the crystal from the crystal structure diagram or cartoon for the protein. So to count the number of amide protons actually from PEPS, we obtain the difference between the molecular weight of the fully folded protein. We subtract that from the molecular weight of the fully unfolded protein, and that gives us the number of amide protons in the protein. So obtaining that information also helps us that when the protein is mutated and we see a change in that number, then we know that yes, different more or less amide protons are involved in secondary structure formation, and then we can extrapolate from there what that means to the activity of the protein and its function in the cell. So in a related experiment, we also try to validate the PEPS by carrying out fluorescence analysis, and we obtained a curve that is almost superimposable with that of PEPS, validating our PEPS experiment. And also we obtained values for CM that were similar to each other. The CM is the concentration at which half of the protein is denatured. So we see a value that is similar to each other. So that validates our fluorescence, our PEPS experiment also. So to conclude, we say that PEPS-HDX compares well with other well-known stability measurement methods like fluorescence. The number of protected amide proton from the crystal structure of wild-type FGF1 and the number of protected protons from PEPS-HDX are comparable. PEPS-HDX is a good method for probing protein secondary structure. It can also be used for folding and unfolding rates of protein, as well as determining the used for protein mapping. Thank you. Special thanks to my advisor, Dr. Jackson Lay, is director of Arkansas State Mass Spectrometry Facility. His assistant is Dr. Rohana. Liyanage is the senior research associate at the facility. And then Dr. Suresh TKS is a professor that supplies all the proteins that are used for my analysis. Is the PI for the biochemistry department, biochemistry lab at the University of Arkansas, Fayetteville. So thank you everybody for listening. I'll take questions if you have them.